Hi, good morning, everyone. Well, thanks for coming despite the snow and the slow traffic. So it's my pleasure to introduce our grand round speaker today, Dr. Sean Davidson. Sean is a professor in the Department of Pathology and Lab Medicine at the University of Cincinnati, and he also serves as the Vice Chair for Research and the Director of the Center for Lipids and Arterial Sclerosis Studies, located at the University of Cincinnati's Metabolic Disease Institute. He received his Ph.D. in Biochemistry from the Medical College of Pennsylvania in 1995 and performed a postdoctoral fellowship at the University of Illinois. He's a member of the editorial boards of the Journal of Biological Chemistry and the Journal of Lipid Research, and he's also a fellow of the American Heart Association. So he also serves on a variety of regional and the national study sessions. His research interests center on the structure and function of HDL. So today he's going to talk about his research titled HDL, a structure platform for cardio and immunoprotective functions. Welcome, Sean.
So thank you very much, Danny. So can everybody hear me okay? Yeah. So thanks for coming out on a morning when it's snowing. If this happened in Cincinnati, I guarantee you people wouldn't get here till 10 o'clock. So we cannot remove snow to save our lives in Cincinnati. So I understand you had my good friend Alan Romali here last week, so hopefully people have been exposed to this idea of lipoprotein metabolism. So I just want to tell you some stuff that we're doing in my lab. I'm kind of a unique person to be in a pathology department. I'm more of a biochemist, a structural biologist, in fact. So I'll tell you a little bit about some of the structural biological work that we're doing in brief. I don't want to put anybody to sleep. And then I'll talk to you about some of the proteomics and the functional and heterogeneous nature of HDL and why I think it's very important for understanding biology. And I don't know how you run this, but I'm happy to stop and answer questions during the session, and we can dynamically readjust the timing of the talk depending on how we talk. So this is just a quick overview of lipoprotein metabolism in case somebody doesn't think about this all the time like I do. Generally, when you're talking about lipoprotein metabolism, you're talking about two organs, the gut and the liver, and then basically everything else, including possibly in some cases the brain. So generally when you take in dietary lipids, these are packaged into lipoprotein particles, which solves the oil and water conundrum in the human body. These things are solubilized and delivered to peripheral tissues and delivering triglycerides and dietary fats as they go along. These chylomicrons then get taken up, the remnants get taken up by the liver, which resecretes cholesterol and other lipids as very low density lipoprotein particles, which eventually becomes the famous low density lipoproteins, which is the primary delivery vehicle for a lot of cholesterol around the human body. And this could be, for example, cells in the vessel wall, around the heart, in the coronary arteries of the heart, for example. And eventually, if you get too much accumulation, especially of oxidized forms of LDL, you get a net atherosclerosis or accumulation, a pathological accumulation of lipid in the arterial wall, and you eventually get atherosclerosis and closing off the oxygen delivery capability of these vessels. So where we think high-density lipoprotein comes in one way is that it actually has the ability to... Did I lose my microphone? Hello? Okay. It actually has the ability to sort of pick up cholesterol from a lot of these peripheral tissues, which... Yeah.
Just move it higher.
Yeah.
Better? Which has the ability to... I'm usually a loud talker anyway, so... has the ability to pull cholesterol out of these tissues. And a lot of these tissues don't have the ability to break cholesterol down unless it's a specific steroidogenic tissue. So we think HDL promotes this idea of reverse cholesterol transport to kind of counterbalance the delivery of cholesterol in, for example, cells of the vessel wall. So this is why HDL has always been thought to be an anti-atherogenic species, and really there's been a lot of study over the last 40 or 50 years about this molecule. It can deliver cholesterol back to the liver, which can then excrete it in the forms of biliary cholesterol or biliary bile acids. So my lab's been very interested in this idea of reverse cholesterol transport, but as you'll see as I go along, there's a lot of other biology that's really important here as well. So this is just a summary of some of the epidemiological evidence that for HDL being protective in cardiovascular disease. You've probably all seen some version of this graph over the years many times. Generally, your risk of coronary heart disease goes up with higher amounts of low-density lipoprotein cholesterol, and higher amounts, or basically goes down with higher amounts of HDL cholesterol. So there's lots of observational epidemiology which supports this. There's been studies that show that lowering LDL levels is good, but if you could bring HDL, or HDL cholesterol is still an independent risk factor after all that is taken into account. There's a mountain of animal and in vitro studies that show that HDL has effects which would be considered cardioprotective. And just recently, a cholesterol ester transfer protein inhibitor drug that was introduced by Merck was able to reduce first cardiac event in humans on top of statin LDL lowering treatment. But this has been a controversial field over the past couple of years because several companies have really spent a lot of money on, especially this idea of CETP inhibition. What this does is basically limits the ability of cholesterol to exit from the HDL particles. So you get a large accumulation of HDL, which makes your doctor very happy, HDL cholesterol at least. But we're finding out that you can increase cholesterol very well with these inhibitors, but it doesn't seem to translate into a cardioprotective benefit, except in that one case that I just told you. So Pfizer spent almost $800 million on a drug that had some off-target effects that actually increased cardiovascular endpoints. Roche had a similar, a little bit weaker inhibitor that basically had no effect in their clinical trials. And I don't have a slide here, but Eli Lilly just recently stopped their trial for no effect as well. So raising HDL in epidemiological studies seems to be a good thing, but when you actually try to do it pharmacologically, at least by this mechanism, it hasn't worked out so well. So I think the reason for that is HDL is an extremely heterogeneous entity, and I'm going to show you evidence of that as we go along. The whole premise for what I'm going to talk about today, and the message I want to try to deliver, is that we've been looking at HDL the wrong way. So you go to the doctor's office, he or she measures HDL cholesterol, and the higher that number is, the better off. The problem is that cholesterol only represents about 20% maximum of the total composition of HDL particles. Most of the lipid in these particles is actually phospholipid. And this is what I think we should be concentrating on. Because I think phospholipid acts as a organizing scaffold, just like it does in the plasma membrane. So for example, a plasma membrane, just a phospholipid bilayer here, acts as a medium where you can get proteins that can diffuse in 2 dimensions and get together and have some new functions. For example, the subunits of a receptor, for example. So you get two proteins diffusing in 2 dimensions. They don't have to diffuse in 3 dimensions. They can get together and have some kind of a new function. We think the exact same thing is happening in HDL. So HDL basically is an extracellular scaffold that allows the assembly of various different proteins to get together so they don't have to diffuse in 3 dimensions, and that comes up with a new function. Okay? So this picture sort of summarizes what I'm talking about. HDL is not just one thing. There's lots of large particles that have certain proteins on them. There's lots of small particles that have certain proteins on them. And I would argue that these are targeted toward very different functions, very different pathologies, and very different biologies. So basically, if you don't remember anything I talked about today, remember this bunch of marbles here. So my lab spends a lot of time thinking about the structure of these particles. So I'm going to tell you a little bit about that work. I know it's 8 o'clock in the morning and there's not a lot of structural biologists in the audiences, but this will give you an idea of why we think these things are such a nice organizing scaffold. And then I'll talk about some of the functional and more clinical aspects of what we're doing. So, and Ling Li is very familiar with this. When I founded my lab, basically I was very interested in understanding the structure of these HDL particles as they go through their life cycle. Okay? So you generally start out with HDL as a lipid-free apolipoprotein, most commonly ApoA1, for example. And it can then, when it interacts with the cell surface, a transporter called ABCA1, you end up with a discoidal or a nascent HDL particle. And then these things can mature under actions of another enzyme called LCAP, which esterifies some of the cholesterol in these particles to make spherical particles. And when I set up my lab, I basically said, well, I want to understand these different structures. But the problem is, as a structural biologist, you want to study isolated and very pure versions of these particles. And with HDL, that's just not possible that you isolate from plasma. So it turns out that we can make a lot of these versions in the test tube using reconstitution techniques. So we can use synthetic phospholipids, we can use recombinantly expressed proteins to generate these particles. So a lot of what I did early on in my career was studying the structures of lipid-free ApoA1, nascent discoidal HDL, and nascent spherical HDL in the test tube. But the idea was always to move toward these real particles that you find, all these different marvels that you find in human plasma. And so these are just different papers that I've published over the years showing the work that we've done on this. But largely we've been successful in applying a series of lower resolution techniques to these proteins all the way through to understand the structure of these particles. And I'll just kind of summarize that very briefly because a lot of it's been published. So ApoA1 is what you would consider sort of the building block of HDL. And it's an interesting protein. It's basically a 243 amino acid protein. It's got a lot of putative amphipathic alpha helices. So it's got an alpha helical segments, about 10 of these. They have a hydrophobic face on one side and a hydrophilic face on the other. And that allows them to sort of solubilize lipid bilayers. And you can imagine this is one molecule of ApoA1 on top here, starting this direction, going around this way. The other one going around in this direction. And you can see this is a model that was from Jerry Segrist's lab. When I think, Ling, were you there at this point?
Not yet.
Not yet, okay. Anyway, Jerry Segrist developed this idea of this double belt model of ApoA1 wrapping around this bilayer of HDL phospholipids. And so our lab wanted to try to confirm this, so we came up with a method of actually developing these in the test tube and then using a technique called chemical cross-linking, where we basically took this thing and treated it with chemical cross-linking agents that would actually link lysine residues with a very specific spacer arm length. Okay? And that's just shown down here. And then you can use mass spectrometry to cut this whole thing up and then actually look and figure out which peptides are actually really close together in the structure. And when we did that, and this is just a two-dimensional representation of this thing, you've got the N-terminus of one molecule going this direction, coming right back around here, the other one going the opposite direction. The cross-linking pattern that we found was very supportive of this idea of this double belt. So these helices form rings that sort of stack on top of each other. And this is just a native gel that shows you can make these things really homogeneous in the test tube. So we move from these nascent particles, which just pretty much have phospholipid protein in them, to a spherical particle. So what we did was we treated these things with a less than cholesterol acyltransferase, which is, I imagine Alan or Molly talked a lot about this molecule. Esterifies cholesterol into a neutral lipid ester, cholesterol ester, which forms a core of these particles, and they sphericalize. Generally, these particles are discoidal. You can see this by a negative stain EM. They kind of stack on top of each other like coins of a diameter of about 100 nanometers. You can make these spherical, which with almost the exact same diameter, but now they have 3 molecules of ApoA1 per particle versus 2. Okay? And you now have this neutral lipid ester core. So we did our cross-linking experiment on these spherical particles, which we can make nicely in the lab. And what we found really gave me a lot of consternation at the time. So we found the exact same cross-linking pattern in the spheres that we found in the disks. And this really confused me for a long time, because how can you have a 3-dimensional structure that has the exact same interactions that a 2-dimensional discoidal particle has? So we were drinking a lot of beer in the lab one night, and one of us came up with this idea that, well, if you take this double belt like I showed you here, with these amphipathic helices sort of colored in different colors, and you put a hinge domain in helix 5 and in helix 10, you can open the molecule up like this. You can then take a third molecule and insert it into that structure to make what we call the trefoil model. And what you can see is that the molecular interactions of this thing are exactly the same as what you would have in the disk version of this thing, right? Which would be this part. But now you have 3 molecules that are interacting the exact same way and gives you the same cross-linking pattern. So now you have a cavity in here where you can have the neutral lipid esters of the cholesterol esters and triglycerides, and then you have the protein sort of making these spines on the outside of the particle. So we published this in PNAS several years ago, and it's interesting. I took this. My family's usually not very interested in stuff I do in the lab, but I took this model home and I showed it to my family, and it's interesting to see how what people see in these models based on what their experiences are. So my son, Jack, he's a gamer. He said, I showed him a version that has 4 molecules. He said, well, Dad, that looks like the Xbox. And we better call it the Xbox model. And my wife, who likes the finer things in life, she says, well, I see the Mercedes-Benz. So, unfortunately, with our salaries, that's the closest we're ever going to get to a Mercedes is on this screen. But the one that scared the hell out of me was my daughter, Sydney. She says, Daddy, I think it kind of looks like a thong. So I haven't let Sydney out of the house since. So all this work was done in reconstituted particles. So what we really wanted to get to was real HDL particles. So again, I'm going to show you a lot of data about how heterogeneous these things are, but one way that you can actually sort of simplify these things is by looking at the main scaffold protein on these things. So I mentioned ApoA1 is the main protein in HDL. There's another one, ApoA2, which is the second most abundant protein. And it turns out that if you immunoseparate or use sulfhydrocovalent chromatography, because A2 has this disulfide in it, you can actually separate particles that have ApoA1 in them without ApoA2, or ApoA1 and ApoA2. This is by no means homogeneous, but there's lots of other proteins on both of these, but at least it was kind of a poor man's way to separate these things out. So what we did was we isolated different density subfractions of HDL using ultracentrifugation, and we looked at the LPA1 versions with this cross-linking technique that I just showed you. And long story short, I'm not going to take you through the whole paper, but this trefoil model that we identified in those spherical reconstituted particles fits very well with what we find in real HDL particles of almost any different size. Okay, some of these particles, like the larger LPA1 2B particles, have 5 molecules of A1, which you can basically generate a version of the trefoil model which does that as well. All the way down to smaller particles which have 3, which would fit with this trefoil idea. So we've actually worked out the structure we think of ApoA1 molecules in a lot of these different size LPA1 particles at least. And this is just a model showing the largest HDL2B particle. This has 5 molecules of ApoA1 on it, with the diameter shown here. And we've actually been able to model this with the correct number of phospholipids. And what you see here is you basically get a ball of phospholipid on the outside. If you can kind of see as it goes by here, you can see there's a cavity on the inside, which is shown in this cutaway here. You've got these amphipathic helices of ApoA1 sort of floating on the surface, stabilizing this particle. And you have this almost basketball-looking thing, this scaffold, which stabilizes these phospholipid bases. And basically everything we can measure experimentally fits very well with this model in terms of the volumes and the components that are there.
So on the internal part,
Yep.
is there a propensity to have hydrophobic residues for the binding of bits on the inside?
Right. If you look at if I showed you, if I had this colored for like amphipathic nature of the helices, the polar region would be on the outside here, so you've got polar amino acids, and the hydrophobic ones are down in here, interacting with the acyl chains. And some of these acyl chains actually curve around underneath the helices.
Thanks.
And then you have this core here where you can have triglycerides and cholesterol esters. So this is really the first, I think, molecularly detailed structural model that we have of what these HDL particles look like. And what's interesting is when you look at smaller particles, this thing's basically collapsed down, but you have most of the surface that's dominated by this ApoA1. So there's a very ApoA1 on the outside forms an interface that can be interface for a lot of different biological reactions. And I'll talk a little bit about that here in a second. So I'm not going to talk about a whole lot of this, but my lab is having a whole lot of fun of using this cross-linking technology to actually understand how proteins interact with these HDL particles. So this is LCAT, less than cholesterol acetyltransferase, I just talked about. We've actually been able to localize a region on these discoidal particles where LCAT is actually interacting. And it turns out that this registry of these ApoA1 molecules can actually affect how LCAT binds to the HDL and affects activity, which I think is pretty interesting. And it may actually, this registry may actually dictate how HDL interacts with other things like cholesterol transfer protein or peroxidase or other things. I won't have time to get into that today, but there's some pretty interesting things going on in the lab. So the main points of the first part is I just want you to recognize that we think ApoA1 is a really important structural scaffold that not only solubilizes phospholipid, but also acts as an interface for other proteins that interact with HDL. It adapts a double belt-like interaction in just about all of its lipid-bound forms. That I didn't get a chance to show you the different sizes, but that cross-linking pattern holds in just about every different size HDL particle we've looked at. And we think these models are helping us to understand the interactions between HDL and its plasma remodeling factors. Okay? So, well, why is all this important? Well, let's move to some of the compositional and functional work that we're doing. HDL is not just structurally heterogeneous, it's functionally heterogeneous as well. So I've talked a little bit about this reverse cholesterol transport idea. HDL in vitro and in vivo can have lots of different functions. So it can affect platelet aggregation depending on the proteins that are present in the HDL. It can modulate apoptosis of, say, endothelial cells. It can actually affect whether LDL lipoproteins are oxidized or not. It can actually detoxify some LDL particles. It has lots of anti-inflammatory roles, both in the immune system and just in the endothelium. It's even been linked to glucose metabolism, so in terms of things like glucose or insulin secretion in the pancreas. And something that's really been interesting to our lab lately is it plays roles in innate immune function. So this functional heterogeneity is actually mirrored by a compositional heterogeneity. So this is a graph that we published, or a figure we published in a review a couple of years ago, where you can, different laboratories have been using shotgun mass spectrometry to look at the protein content of isolated HDL particles. And generally, my lab keeps a spreadsheet online that keeps track of all the different labs that have done this. And basically, every protein that's been found in at least 3 different laboratories is listed as an official HDL protein. That number is almost 100 different proteins right now. And I've just listed these out here, and you can characterize them in different ways. So you have the traditional lipid metabolism and transport, all the APOs that you traditionally would associate with HDL. The things that are involved in blood clotting, hemostasis, lots of proteins that are involved in immune response, including a lot of complement factors. You have sort of exotic functions like metal binding that can go on, or metal binding proteins, even vitamin transport. And these proteins can be organized in different ways. So you have a lot of that are involved in acute phase response. Protease inhibition is a big theme that you'll find in HDL proteins, and then complement factors. So you can characterize these different ways, but the point I want to make is the traditional lipid transport functions actually makes up a minority of the proteins that you find associated with HDL. So HDL is not just one thing. There's lots of different particles that have these different proteins on them that I think are probably executing different aspects of biology and pathology. So we sort of set about thinking about how can we try to separate these particles to understand how different proteins associate with different functions. One of the things we settled upon was the most mild separation we can think of, which was gel filtration. So this is just a trace of a gel filtration size exclusion technique that we have set up in the lab, where you have large stuff on this side, small stuff on this side. So you can just take human plasma, separate it on what we call a triple superdex gel filtration system. Things with ApoB in them, like LDL and VLDL, show up down here. And if you just look at just pay attention to the phospholipid trace for the red one here. You get VLDL and LDL here, and then we optimize this system to try to spread the HDL particles across, at least as defined by ApoA1, across as many fractions as we could. You can see that that's stretched out here. And then you have things like small lipid-free proteins like albumin and things like that down this end. So we said, okay, this is a really gentle way to separate these particles, and we wanted to start to use this to try to understand how these different proteins are segregating across size species. And I could show you 20 graphs that look just like this, but this is just I've kind of switched up the axis on you. This is the large stuff down here, small stuff down here. But basically we just take the mass spectrometer and we take fractions from the separation and we just look at where proteins are eluting using the mass spectrometer. You can see ApoA1 covers this whole range. This is the HDL range generally. ApoA2 covers that range, but what you see is a lot of these other ApoA proteins are actually differentially distributed across this size pattern. You have some that are really concentrated down in the small particles. You have some that are really concentrated in the large particles. And this, again, is more evidence that HDL is just not one thing. It's a whole family of different particles that have different compositions. So this is kind of a cartoonized version of a lot of work. But we decided, okay, we're looking at the protein composition across these fragments. Why don't we look at individual functions of HDL? So these are 3 of the main functions that everybody sort of associates with HDL. The ability to protect LDL from oxidation with a pro-oxidative insult. the ability to promote cholesterol efflux from cultured macrophages, for example, and then an anti-inflammatory function, which is an endothelial cell adhesion molecule expression assay. And what we found was that across these size ranges, these different functions sort of localized to different fractions, different size HDL particles. So pretty much cholesterol efflux was sort of concentrated across anywhere you found phospholipid in the blue here. But things that are involved with protection from LDL oxidation seem to be concentrated down here in much smaller particles. Whereas the anti-inflammatory effects seem to be in sort of medium-sized to small particles. So this is, again, evidence that not only do you have structural heterogeneity in terms of composition, you've also got a lot of functional heterogeneity. And our dream is to sort of parse these things out and try to figure out what particles are doing what, in the hopes that we can do better than what the pharmaceutical companies have been doing by raising all the HDL, at least in terms of cholesterol. Can we raise specific subspecies that are more important for particular disease states? And combining our proteomics with these functional assays, we've actually got ideas of proteins that we sort of associate with these different functions, and these are things that we're working on confirming now in the laboratory. Interesting, cholesterol efflux, the biggest factor that we found that correlates is just the amount of phospholipid. It doesn't seem to matter the amount of protein that's, or the type of protein that's there. So about this time, I was given a talk in Cincinnati, locally, and a resident or a fellow, clinical fellow at Children's Hospital of Cincinnati, which is right next door to us, came up to me and said, well, I have a bunch of adolescents that I'm studying, and I'd really like to see if their HDL profiles change with diabetes or obesity. So she basically came to my laboratory, and we basically analyzed her cohort. And this, I'm not a huge clinical operation in my lab, but we do small runs on human patients. And what we found was that if you take this individuals that are lean, individuals that are obese but don't have type 2 diabetes, or individuals that have type 2 diabetes by the clinical standard, we actually saw a shift in their HDL pattern when you look at it by this gel filtration assay. So this is a lean individual here. You get a fair amount of these larger HDL particles. We call this fraction 21 and 22, which are these larger HDL particles, and there seems to be less of these smaller ones here in the green. But as a person becomes obese, these larger particles seem to drop down, and then we have full-blown type 2 diabetes. And again, these are youth, about 18 to 21 years old. You completely lose these large particles, and you get a little bit more of these smaller particles. So we were really interested in this, and Amy Shaw, who is the clinical fellow that I told you, because at this point we got this result, and she basically moved into my lab. Her entire operation moved into my lab, and she's interested in looking at these parameters as they relate to clinical readouts of vascular health. So these are youth. So what she's been doing is using something called pulse wave velocity. I'm sure a lot of you know more about this than I do, but basically it's a way of measuring arterial or vessel stiffness. So you wait for a heartbeat, you can measure, have a cuff on the neck, you have a cuff down the leg, and you can measure how far that pulse from that heartbeat translates between two different points. And basically if that happens very fast, you have very stiff arteries. If it happens slower, you have patent arteries. That pulse takes longer to get down to this point. And it's been shown in adults and in adolescents that the stiffer the arteries are measured by this, there's a high correlation with eventual atherosclerotic disease that shows up. And so what we did is we took this data that we had in all these kids and took this data, and we actually started correlating things with this pulse wave velocity. So if you look at LDL cholesterol, which is the sort of gold standard that everybody looks at when you look at prediction for atherosclerosis, we really didn't see much of an effect. So this is, I can't remember what, this green is the normals, red is type 2 diabetics, and blue are the obese, and you can see there's really no prediction for this pulse wave velocity from the LDL cholesterol. HDL was a little better, but still it wasn't significant. But if you look at this fraction 22, you remember from this fraction right here, okay? The amount of this large particle seemed to be very highly correlative with the pulse wave velocity. So type 2 diabetics had really low amounts of this fraction, but had the more stiff arteries. Obese were sort of in between, and then the normals had much lower. So this particular subfraction of particles actually was much more predictive than the traditional clinical biomarkers that we have available to us now. So Amy has gone on. We have, unfortunately, in Cincinnati, we have a large problem with childhood obesity, which is happening more and more across the country. So we have a clinic where kids 18 to 20 years old are coming in for being morbidly obese. and they're getting bariatric surgery. Okay, so this is vertical sleeve gastrectomy, where basically they just cut off a pouch in the stomach, and you now have a much smaller stomach area, and reduce food intake, and these kids lose weight very quickly over the next year or so. And I'm sure you've heard a lot of their insulin resistance phenotypes go away even quicker than the weight loss can happen. So we actually looked at these kids that are getting before and after bariatric surgery. This is after a year, and we did the same separation here. So this is a lean control population here, and you can see those large particles in fractions 21 and 22 are there. The baseline kids before surgery, and as I said, they're very obese with high BMI. They have very low amounts of this large particle. What's interesting to us is that this is 1 year after bariatric surgery, even though they're still considered obese from the other experiments that we showed, we've got a recovery of these large particles after a year. And we've actually shown that's happening as fast as 6 months and maybe even 3 months after the surgery. So these are tracking along pretty well with the weight loss. What's interesting is that these things can actually recover, even though they're obese, almost to what the lean phenotypes look like. And we've done a little bit more work where we've actually taken different subjects that are lean with insulin sensitivity. So these are normal, healthy kids. You can have lean kids that are insulin resistant by the typical clinical definition. You can have kids that are so-called healthy obese, so they're obese but they're insulin sensitive. And then you can have obese, insulin resistant, and then full-blown type 2 diabetes. So we actually took cohorts of all of those, and what we see, I won't belabor this point too much, but if you look at this fraction 21, basically you get a continuum between lean and type 2 diabetes in this fraction 21 where they're going down. And we actually were able to sort of statistically parse out the factors that seem to be most important in this large subfraction. Turns out that whether they're obese or not seems to have a big effect, and then whether they're insulin resistant type or diabetic has this contribution. So something's going on with inflammation or meta-inflammation from accumulation of adiposity that's affecting the distribution of these particles. So we're very interested in developing, so the problem with this assay is it takes about 3 hours to run this gel filtration separation. This is nothing you'd ever want to run clinically, right? So what we're doing now is we're doing a lot of work proteomically to try to figure out what it is about these particles that's interesting. Can we develop an ELISA or an immunoassay that can help us detect this thing very quickly in a clinical setting that might help us have better prediction, at least in youth populations for kids that are at risk for cardiovascular disease. So how's this working? Well, one idea that we have at this point is that these large particles, and I won't have time to show you the data, but one of the main features of these large particles is they have a lot of apolipoprotein called ApoE on them. Okay, ApoE is interesting because it's got a lot of binding sites for a lot of interesting things in plasma, including the LDL receptor and proteoglycans in the plasma in the vessel wall. And we were intrigued by this study that was published previously from Herman Camejo. What they found was that people with large HDL, this is called HDL2, this is small HDL, HDL3, people with high amounts of HDL2 actually have less LDL that can stick to proteoglycans. Okay, so you have these large HDL particles, we think that they're competing with LDL sticking in the vessel wall to these proteoglycans. So that might be a healthy phenotype to prevent LDL from accumulating in the vessel wall. HDL3 and the smaller HDLs doesn't seem to have an effect. So we think that this particle that we're looking at has ApoE on it. It may be competing for LDL sort of accumulation or inappropriate LDL accumulation in the vessel wall. And that's kind of a working hypothesis we're on right now. If anybody has a better idea, I'd love to hear it. So, okay, so that's sort of one aspect that we're doing. Another thing that, and since I know you guys were, or some of you were interested in the proteomics, I also wanted to show you some proteomics work that we've done by separating lipoprotein subspecies based on their scaffold protein. So this ApoA1 versus ApoA2 that I told you about. And our hypothesis is that the presence of A2 influences the complement of other of these so-called client proteins or associated minor apolipoproteins with these particles, and that can alter HDL function. So we have, and again, we're doing small studies in the lab, but basically we have 4 male donors, and we took plasma. These are just normal lipid donors. We isolated particles from plasma, and we isolated also pre-isolated HDL by ultracentrifugation. And then we ran them down two different antibody columns. The first is an anti-ApoA1 column, and basically what flowed through that, we put onto an ApoA2 column, and we're basically able to isolate LPA1 and LPA1A2. A slightly different method than I showed you before, but it works the same. One thing we looked at was, well, different methods for isolating HDL. How big of a difference does that make on the proteome? So we actually isolated this in two ways. We isolated, we took plasma and we isolated HDL by ultracentrifugation first, and then we also isolated by just the ApoA1 content. And when we looked at the proteomics, and I'm not going to go through all the mass spectrometry, what we found is that if you separate, if you define HDL by its ApoA1 content, you get a lot more proteins using this method than when you spin these things for hours at a time in high salt gradients, which is the typical method of isolating these things by ultracentrifugation. So there's relatively few proteins that we can consistently find in ultracentrifuge samples. So we think that this ultracentrifugation spins off a lot of different proteins that could be very important biological. A lot of these minor proteins that I showed you that have some of these more exotic functions, for example, were no longer present. So one of the things I've been advocating for is to try to come up with more gentle ways of separating out these HDL particles for biomarker studies in the future. So anyway, getting back to the LPA1 and LPA12 particles, this just shows you we did our job well. So this is we separate the particles, we look at them by SDS gel electrophoresis. So ApoA1 is right here, ApoA2 is right here. You can see it's completely missing in the LPA1 particles, but it's there in the LPA1A2. And this is just different ways of showing that. This is actually a Western blot. We were actually able to get rid of ApoA2 by Western blot, which is pretty amazing. And when you look at these by native gel electrophoresis, so this is regular HDL separated by ultracentrifugation, you sort of get this smear on a native gel from particles between, say, 8 and, say, 12 nanometers in diameter. The LPA1 actually broke out into two very distinct subpopulations, and LPA1 and A2 is sort of a smaller smear that encompasses these here. And it doesn't matter if you separate that by ultracentrifugation or just directly out of plasma, you get the same results. And this is just a gel filtration analysis of that same thing. Well, but what we're interested in is looking at the proteome between these two different populations. So what we found is this is isolated from plasma using the antibodies. We found, 88 some proteins in these particles, and a lot of them were overlapping between LPA1A2 and LPA1. But most of the protein diversity was actually in the LPA1 containing particles. So it seemed like when ApoA2 is present, you get a lot less proteins present in that particle in terms of diversity. And by the way, this difference completely disappeared when you separate particles by ultracentrifugation, which is what a lot of people have been doing for 30 or 40 years. And then we looked at just a simple gene ontology analysis of the proteins that were found in these different fractions. And what we found is that the LPA1A2 particles seem to be associated with things like you would typically associate HDL with, like lipid transport, lipid metabolic processes. But the LPA1 particles had proteins that were, a lot of these proteins involved in homeostasis, hemostasis, sorry, metal ion binding, complement activation, and so forth. So a lot of these more exotic functions of HDL seem to be concentrated in the LPA1 fraction. And I'm not going to have time to show you some of the functionality of this, but this was actually, we did some cholesterol efflux assays with these two different particles, and basically LPA1 seems to work better in terms of cholesterol efflux, LPA1A2, I'm sorry, which is consistent with this idea that it's involved in lipid transport. The LPA1 was not so good at that, and we're trying to develop other assays to test out some of these different functions in the laboratory. So the point I want to make is that not all HDL are created equal. We think that HDL, if we can biochemically subspeciate these things and actually understand what their functions are, we think that we could better target these molecules better than what the pharmaceutical companies have been able to do so far. So if you can think about it, if you have two patients, one that's healthy, that has a HDL cholesterol level, this is a male, has an HDL of 45, which is a reasonable amount of HDL cholesterol, but you also have a person that has documented cardiovascular disease that has the exact same HDL-C level. But if you were somehow magically able to spread out and figure out what all these different subfractions are, you might see that there's dramatic differences here. There's the cardiovascular disease person might have less of a healthy type of an HDL particle and more of a detrimental or dysfunctional HDL particle. And if we can actually target what these subspecies are, we might be actually be looking at different subspecies that are performing different functions in biology. And the idea is we could target those, not only measure them and use them as biomarkers, but hopefully you can get to a point where we can raise those pharmacologically to actually have an effect on the disease state we're looking at. And this may work for cardiovascular disease, but I'm also hoping it will have effects on other functions that have been identified for HDL. Okay, so how are we doing on time? So I know some folks are interested in our work on HDL and the immune response. So I want to show you one kind of crazy result that we found, and hopefully some folks in the audience will help us out with this, because I'm not an immunologist of any sort. But HDL, as I mentioned, it has roles in innate immune function, and it always seemed to me that since cardiovascular disease tends to strike after reproductive age, HDL probably didn't evolve to protect us from heart disease. It evolved for something more basic, right? And probably most evolutionary drivers have to do with host defense, or a lot of them do. And there's lots of evidence for this. There's actually a subfraction of HDL called trypanosome lytic factor. Okay, this has a protein called ApoL1 in it, and another one called haptoglobin-related protein. This is actually a very specific subfraction of HDL that's been identified. And what happens is that this trypanosome lytic factor can actually get in and kill trypanosomes. Okay, this is a pathogen that can, in certain species, can cause African sleeping sickness and other diseases. What happens is this HDL particle has these two proteins on it. It actually binds to a receptor at the base of the flagella of the trypanosome and actually tricks its way into the cell. And then ApoL1 is a lytic protein that can actually poke holes in the membrane from the inside out, killing the organism. So this is something that's completely not involved with lipid transport at all, but it has a very specific function in terms of host defense. So there's a buddy of mine in pathology at UC. We have a very varied department. He studies Aspergillus fumigatus. This is a fungal pathogen that we're all breathing in the canidia for right now. This is in soil, it's in compost, it's all over the place. It's an opportunistic fungal pathogen that can actually become a problem in people that are immune compromised. So if they have solid organ transplants, for example, and your immune response is compromised, you can get this filamentous fungal growth in the lung that can actually, once it gets past a certain point, the prognosis for these folks is really, really dire. So I've been listening to David, his name's David Askew, I've been listening to his talks for many years. I'll stand up and talk about lipids, he'll stand up and talk about Aspergillus, so we really didn't have much in common. But eventually I started thinking about, well, is it possible that HDL may actually play a role in the susceptibility to this fungal pathogen? And this is just one of those happenstances that two people in the room were together at the right period of time. So I said, David, what if we took an ApoA1 knockout animal, which has very low amounts of HDL, and just see how susceptible it is to your fungal system? So what he does is basically he takes mice and he spritzes these canidia into their nasal passages after going through an immune suppression regimen, and then basically lets them either clear the organism or not, depending on what treatments and things he's looking at. So we basically took wild-type mice that were immunosuppressed and were either given this spritz of Aspergillus spores or not. And these mice typically have HDL cholesterol levels that are about 60 mg per deciliter. We took the knockout animal, which has no ApoA1. This is a whole body knockout. These were immunosuppressed as well, but they have much lower amounts of HDL cholesterol, and of course no ApoA1 in their HDL particles. So this is just a survival curve of the experiment, 100% survival versus 0 over the days of the experiment. So basically, if you just take a C57 Black 6 mouse, generally you put them through this protocol. They're immunosuppressed. You spritz in the canidia, and they generally can clear it if you let the animal alone. Okay, so most of these animals were surviving. There was actually one animal that died over 2 weeks. Same thing with the ApoA1 knockouts. They seem to do fine. And then sham, we did a sham, which is PBS rather than the canidia. But what was really interesting, when we looked at the ApoA1 knockouts, these animals basically were highly susceptible to this organism. Okay, and they died within 14 days. Most of them died within 14 days, and really due to a massive inflammatory response in the lung. And David was floored by this. He said he'd never seen anything having this big of an effect. So again, I'm not a pathologist, but we did a little bit of pathology here. So this is these are lung sections where we looked at the ability of ApoA1 to affect the burden of the fungal burden in the mouse lung. So this is a GMS stain, which basically gives you an idea, and this is not showing up very well, but basically if there's little black dots here. That's the organism in the wild type that actually is spritzed in. And over 3 or 4 days or so, a normal mouse will actually be able to clear this, so you don't see any black here. This is an H&E stain to give you an idea of the inflammatory response. You can see that that's actually it's pretty normal after day 3. The ApoA1 knockout, you see a little bit of black spots here, about the same, but then much more of the fungus stays around after day 3, and you get this massive inflammatory response, mostly neutrophils and things like that that are coming into these sites in the lung. And if you look at the actual amount of fungus that's in the lung, using a marker, the beta-glucan is a part of the cell wall of the fungal pathogen. You can see that the wild type, you start out with a little bit and it kind of clears it over 3 days, but you get a large amount of the fungus actually taking off in the ApoA1 knockouts. So we've done, and there are several experiments that I won't have time to show you here, but basically we think that ApoA1 is actually helping the innate immune response in these organisms. So if you take macrophages that are in culture and you treat them with or without ApoA1, we can see that the macrophages that have ApoA1 in there seem to do a better job at clearing these canidia, at least in vitro. And we think that this may actually be happening in the lung. You need to have ApoA1 and HDL around in order for the macrophages and neutrophils to clear the organism properly. So our best hypothesis at this point, I don't want to spend too much time on this, is that we think ApoA1 and maybe HDL coming in from the periphery can limit fungal burden by stimulatory effects on the host cells, and by adjusting the host response to promote clearance without this runaway hyperinflammation. So the mice actually don't die from the fungal pathogen. They seem to die from this hyperinflammatory response that we haven't gotten a complete hold of yet, but we think HDL can sort of temper that. So, it's kind of crazy, but the pharmaceutical industry is spending billions, literally billions of dollars on HDL cholesterol-raising therapies targeted toward cardiovascular disease. And I'm wondering, maybe not CTP inhibitors, but could some of these drugs work for things like aspergillus infection in pre-transplant patients, for example? And this is some of the ideas that I've been pitching with this limited data to pharmaceutical companies. Okay, so with that I'll stop. But I have a great group of people in the laboratory. This is Amy Shaw, the clinician that I told you about, who's basically moved her operation into my laboratory. A lot of the structural work that I showed you was done by John Melcher, a very talented postdoc in the lab. A lot of different people contributed to this, including Frank Sachs, a collaborator of mine from Harvard, who's helped us out with a lot of the proteomics stuff. So with that, I'll stop and be happy to answer any questions you might have. Figured you'd have your hand up.
Very exciting work. I was really struck by the data you were showing that the larger particles of the HDL particle was correlated with the stiffness, basically the arterial. There's something actually I want to do in mice, for a different reason.
It's interesting, just a comment before you ask the rest of your question. If you look at that same trace by cholesterol, which is what everybody does,
right.
That difference in those large particles is much, much more limited. When you look at it by phospholipid, which is all the traces I was showing you, that difference just jumps out at you. It's amazing. So
exactly,
it's a matter of looking at the right analyte in these different particles.
Right. So basically, the larger particle is caused by more phospholipids on that particle, based on your data. So you mentioned about the possibility of the ApoE. That's actually where I'm going. If I recall, if you can go back, one of your data was showing the different size of particles, and then with all the apolipoproteins in there, your proteomics data. I didn't actually see the ApoE. That was the question I was.
Oh yeah. So
yeah.
You're talking about the
one you had a plot. Yeah, this one.
Yeah.
And I was looking at that, and then I didn't. Yeah, because I was looking.
Ordinarily, when I have more time, I have another slide that basically shows you a heat map with all the proteins we looked. 100 and some proteins, and I've got that data. Generally, you'll see. Let's see, get myself oriented. Generally, ApoE shows up, in the larger fractions of
larger,
the HDL.
okay.
And that's
Yeah.
been known for many years.
Okay.
It seems to like a large surface area.
So I was also wondering, the difference you saw, did you also look at CTP in those folks?
CP activity? Or
The activity, yeah.
no, haven't. I think Amy's working on that now, but we don't have data on that yet.
Good.
Because, yeah, I mean, that's one of the things we'd like to understand is what metabolic process is resulting in this large particle. And CTP is an obvious choice. We're looking at, lipoprotein lipase and things like that. What's interesting is we've looked at circulating factors, and one of the things that seems to correlate with the presence of that particle is adiponectin, which is a secreted factor by adipose tissue. So we don't know how that relates yet.
Thank you.
So as long as we are on this particular diagram, I was wondering whether you have looked at the ApoC3, ApoC2. I see that I can't tell which is which.
C1.
That's C1 and C2, I guess I just threw on this one, but yeah.
C2, yes.
And
I
the
can't-
C3 is the green one? No.
Yeah, this is C2 is the green, so I'm guessing, I think it's that one.
Yeah.
And the C3 is the...
Is C3, oh, I'm sorry,
Yes,
yes.
purple.
Yeah,
Purple.
that's
Right.
this one here, so. Yeah, what we've noticed, and now that I remember, is C1 and C2 seem to have similar patterns. C3 sometimes is different. And, I don't know if you're familiar with the work that Frank Sachs has done, but C3 seems to have very interesting effects on HDL. When it's present in HDL, those HDL particles are not nearly as associated with cardiovascular protection than the ones without the C3.
In fact, we have published that in the MESA study recently.
Oh, yeah, okay.
And in fact, when Alan came, Alan was talking about concentrating on C2, which is obviously related to C3.
Right.
But you haven't looked at these in correlating to disease states. C3
In terms of like levels or distribution? No.
distribution.
I mean, one of the things we're doing now is looking at atherosclerosis versus non in as well-defined populations as we can. I'm not happy with the reproducibility of measuring the amounts. We're trying to quantitate these things using isotope dilution. And I think there's a lot of people publishing on this that haven't really worked out the reproducibility and the quantitation of these things yet. So, no, we haven't looked at that yet.
Thank you.
Hi.
Leo.
Go ahead.
So a question on the kids study you showed with the bariatric surgery, where after a year they may still be obese, but they have the elevated fraction 22 or
Right.
whatever it
Yep.
was that you showed. What can you how can you explain that, or is it we just have to wait more time for them
Yeah.
to not be obese, or why doesn't that change
Yeah, I don't know.
over time or
one-
something?
We had thought it was just simply the amount of adipose tissue
Yeah.
seems to correlate with this fraction. But that's definitely, and that experiment shows that's
It's not
definitely
the
not the
case.
case.
Right.
So maybe it's the rate of adipose loss or something that's affecting the HDL particle metabolism. But it's a great question, and I would love to be able to figure out a way to manipulate. I mean, that's the ultimate goal. First of all, this is all correlative, right? So
Sure.
this particle seems to correlate with vascular health. We don't know if it's a causative relationship or not, but eventually down the line, we'd like to analyze this particle and correlate it better in a much more higher throughput clinical setting. But there also may be a causative effect there, and we'd obviously like to understand the metabolism behind how this is being formed, and just not there yet.
Thanks.
I think you've been waiting. Sorry.
Yeah.
Go ahead.
Question?
Green shirt. Or? Go ahead. Yeah.
So have you looked at the HDL profile and the fraction change after immunosuppression? You could tell the either mouse or the human with immunosuppression or without.
Yeah. So he's using a steroid for the immunosuppression. And we have done that. I'm trying to remember what the data was. The profile does change, and I think the HDL levels drop a little bit in the wild type mouse. But I don't remember the exact distribution changes.
It was very interesting, compelling data about the relationship with the immune system, potentially. And I think your first slide showed liver, the guts,
Yep.
liver. And it's important to remember that lipids, I think if I recall, that's very remote for me, travel different way, and they channel through lymphatic system
Sure.
and to thoracic duct. Have you had a chance to look at the lymphocyte composition in mice who have no ApoA,
No.
do they have differences in lymphocyte subset and trafficking?
No, that's a great idea, and that's definitely on the drawing board for doing that, yeah.
Related to the question about the obesity, if they lose weight, do they become more active and are exercising more? What is the exercise effect, by the way, on your analysis?
We have not looked specifically at exercise. I mean, generally, you know, from what I understand from Amy, a lot of these kids get the surgery, you know, they're put on a dietary plan and I assume an exercise plan as well, but a lot of them don't stick to that long term. So it seems to be the rate of change of the adipose tissue, but we haven't done a specific exercise study. Generally, exercise is thought to be beneficial for total HDL cholesterol levels, and we haven't looked at the effect of. Now, I can tell you, I'm on a cycling team, and I had a couple of my buddies come in that are really, really healthy, you know, older folks like I am, and they generally tend to have larger amounts of this large particle. That's a very unscientific study at this point, but you know, we'd like to think that that's correlating in adults too. Yeah.
In relation to that comment, many clinicians, going all the way back to the Framingham study, have been hooked on the cholesterol level in making therapeutic decisions for their patients. Should we switch to be looking at subsets of HDL?
Yes. and this is why the NIH should just do away with this whole silly grant writing process and just fund my lab, because you know, we're one of the few people that are really taking a hard look at the different biochemical ways you can try to separate these particles. They have a lot of the same physical chemical properties. That, you know, a lot of these proteins are moving back and forth in these HDL particles. It's going to take some time and some skill to separate these things out, pair them with specific functions, and try to target those things. But I think it's really important, not only for cardiovascular disease, but you know, chronic inflammatory conditions like rheumatoid arthritis and things like that that HDLs had links to, and maybe the immune.
Is that going to be available on a simple basis in clinical labs pretty soon?
That's our goal. I mean, you can't do a 4-hour gel filtration separation and then follow that up with mass spectrometry in each bracket. So, you know, we were working with people that specialize in antibody generation to try to, because we found that the ApoE conformation in these large particles actually differs from ApoE in conformation, at least by limited proteolysis and things like that in LDL and VLDL and even the smaller HDL fraction. So I think there's some immune targets we can go after in these things. It's just a matter of getting it done. But that's the goal. I'd love to have an ELISA that will point to these large particles.
Thank you, Sean.
All right, thank you.